Hello friends, welcome to my channel Easy Life Science. Here I will be talking about starch hydrolysis test or amylase test and lipid hydrolysis test or lipase test. So please subscribe my channel Easy Life Science. Like my video, share it to your friends and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. A starch hydrolysis test, or it is also known as a amylase test. The many bacteria produce the extracellular enzyme in a class hydrolase known as amylase. The hydrolase enzymes catalyze the splitting of an organic molecule into a smaller molecules in the presence of water. The amylase is one of the enzyme in a class hydrolase, and the substrate for the amylase enzyme is a starch. And the starch molecule consists of two constituents. One is amylose and another one is amylopectin. The amylose is a unbranched glucose polymer made up of 200 to 300 units of a glucose, and a glucose units in a amylose are joined by a alpha 1,4 linkage. The amylopectin, it is a large branched polymer and it is with a alpha 1,4 and alpha 1,6 linkages in between a glucose units. Both amylopectin and amylose are rapidly hydrolyzed by a certain bacteria, for example Bacillus subtilis, and a fungi like Aspergillus niger. Using the amylases or alpha types of amylases and yields the dextrins, maltose and a glucose. The starch, it is made up of amylose and amylopectin. It is a large polysaccharide. By the action of bacterial amylase, in presence of a water molecule, the starch get hydrolyzed into a dextrins, maltose, it is a disaccharide, and a glucose. The interpretation to perform this experiment, the media used is a starch agar medium, and in a medium there is a starch as a substrate for the activity of a amylase, and a starch gives a blue color in presence of a grams iodine or a iodine. When the iodine comes in a contact with starch, it forms a blue to brown color a complex. But hydrolyzed starch, when it get converted into a dextrin, maltose or a glucose, this hydrolyzed starch do not produce such kind of a blue or a brown color in presence of a iodine. If a clear area appears after adding grams iodine to a medium containing starch and a bacterial growth, we can say that the clear area is of a zone of a hydrolysis of a starch, and it is because of action of enzyme amylase produced by organisms. The amylase has been produced by a bacteria. If there is no clear zone of a starch hydrolysis, then we can say that the given organism does not have a ability to produce enzyme amylase. The medium used for this practical or experiment is a starch agar plate, and the starch agar plate, it is nothing but a modification of a nutrient agar. It is a simple nutritive medium in which there is addition of a starch. The nutrient agar is with a beef extract, pancreatic digest of peptone, which provides nitrogen, vitamins, carbon, amino acids. It is also with agar agar powder, and this agar agar powder is a solidifying agent, and the starch, it is a substrate for the activity of a enzyme amylase produced by an organism, and the starch is a carbohydrate. The composition of the starch agar medium, and this composition is for 1 liter of water. Peptic digest of animal tissue 5 gram, sodium chloride 5 gram, yeast extract 1.5 gram, beef extract 1.5 gram, starch 2, agar agar powder 15 gram, and pH 7.4. This is a composition for a preparation of starch agar. After preparation of starch agar, plates are sterilized. Then the procedure or a method for the amylase detection test by using a sterile technique, that means at a aseptic condition, by using sterile wire loop, streak inoculate the test organism into a center of labeled plate, or we can say that a spot inoculation. One can use a positive sign manner inoculation for this test. After inoculation, incubate the bacterial inoculated plate for 48 hours at 37 degrees Celsius temperature. After incubation, flood the plates with a iodine solution for 13 seconds. Pour off the excess iodine. Examine the clear zone around the bacterial growth. If there is a clear zone around the bacterial growth, this clear zone is a zone of hydrolysis of starch. Expected results. A positive test is shown by a clear zone around a line of growth after addition of iodine solution, indicates that the organism has hydrolyzed starch. The negative test, a blue, purple or black coloration of a medium, depending on a concentration of iodine, shows that a test is negative. Here in these two images, the first image shows a starch hydrolysis test positive, and this shows a starch hydrolysis test negative. Here is a growth of organism or a growth of inoculated organism, and this is a clear zone or this is a colorless zone. But in this negative test or negative plate, there is no any zone of hydrolysis around the growth. Intact medium is blue colored, but here a colorless zone around a growth of organism is observed. So this test is positive and this test is negative. Means during a growth, organism produce a enzyme amylase, and this produced enzyme by the organism acts on a neighboring starch, and the starch get converted into a glucose. So it gets diminished from the medium. So it does not get reacted with the iodine and appears as a colorless. Then the next test, a lipid hydrolysis test. The lipids are high molecular weight compounds possessing a large amount of stored energy. 1 gram of lipid gives a 9 kilocalorie energy after its breakdown. Two common lipids catabolized by bacteria are triglycerides, or these are also called as triglycerols, and phospholipids. These two lipids are commonly catabolized by bacteria. Triglycerides are hydrolyzed by enzyme called as a lipase. This lipase acts on a triglyceride and converts it into a glycerol and a fatty acids. Glycerol and a fatty acid molecule can then be taken up by a bacterial cell and further metabolized to a number of reactions such as glycolysis, beta oxidation pathway, and TCA cycle. These lipids can also enter other metabolic pathway where they are used for a synthesis of a cell membrane phospholipids. Since the phospholipids are functional components of all cells, the ability of a bacteria to hydrolyze host cell phospholipids is an important factor in a spread of a pathogenic bacteria. When a lipase producing bacteria contaminates a food, this lipase producing bacteria acts on a lipid present in a food, and this lipid get converted into a butyric acid, and this is known as a rancidity. It is a one kind of a food poisonage because of a lipase producing bacteria. This bacteria contaminates to a food, causes a poisonage of it, and rancidity is a term from it. Term for it. The media used for this experiment is a media with a tributyrine. It is a substrate on which there is a activity of a lipase. After degradation of this compound, the compound gives a clear zones around a growth of lipolytic bacteria. Otherwise, if the organism does not have a lipase producing ability, the medium appears as intact. Media for this test is a tributyrine agar. The composition of it, a peptic digest of animal tissue, yeast extract, and agar-agar powder. Procedure: inoculate the tributyrine agar medium with a single line sticking of organism. After inoculation, incubate the plate at anaerobic condition in a gas pack and maintain a temperature 35 to 37 for 24 to 48 hours. For anaerobic organism, and for aerobic organism, incubate the plate for 35 to 37 degrees Celsius temperature for 24 to 48 hours. After incubation, examine the plates for a growth of organism and around the growth for a presence or absence of a clear zone of a lipolysis. If the organism has the ability to produce an enzyme lipase, you can see for a lipolysis zone around the growth. This lipolysis zone is a clear zone. The interpretation of the test, the positive test when a formation of a clear zone around a bacterial growth. This shows a positive test, whereas no such clear zone around a growth shows a negative test. Application of this test helpful in identification of a bacteria that secretes a lipase, including a member of a family Enterobacteriaceae, Fusobacterium, Propionibacterium, Clostridium, Pseudomonas, Mycoplasma, Corynebacterium, and Staphylococcus are lipase positive organisms. This test is also useful for detection and enumeration of a lipolytic organism in a food and other material which are responsible for a food spoilage. So, thank you for friends. Thank you for watching me. So please subscribe my channel, like my video, and share it to your friends. And don't forget to press the bell icon so you'll get the notification of my newly uploaded video. Thank you once again.